Welcome to the M22 Topic 2 revision sessions. My name is Caleb. You can just call me Junior or whatever. I don't really care. We'll be covering most of Topic 2. I won't be covering everything past 2.5. I will be doing that in a separate section just because, for one, there's 9 subtopics to Topic 2, so it's easily the longest one by far. The sessions might get way too long for you guys if I end up doing all of them. And the last 4 are core, they go along with topic 7 and topic 8. So I'll be covering 2.6 to 2.9 with topic 7 and topic 8, because topics 7 and 8 are fairly short. And if I end up doing 2.6 to 2.9, it might end up becoming repetitive if I do when I do topics 7 and 8 separately later on. I don't think I have to remind anyone to keep their mics muted. Seems like everyone's doing a great job. So with that being said, I will start now. We're covering molecular biology. So what exactly is molecular biology? So molecular biology has to deal with the structure and function of macromolecules and the basis of molecular activity involving mechanisms, synthesis, and modification. So there are going to be 4 main classes that you have to talk about. There are carbohydrates, there are lipids, there are nucleic acids, and there are proteins. A lot of carbohydrates are used for cell communication structure, and they're components of our DNA and RNA molecules. Most of the lipids are going to be energy storage. They're better energy storage. You can find them as steroids, which are a type of hormone. Nucleic acid, DNA, RNA, easy enough. And proteins are associated with catalyzing reactions and the transductions path. So different macromolecules have different components. Monosaccharides will form together to make disaccharides, and then polysaccharides. Many of them form into a ring structure. Think of ribose and deoxyribose, and most of them can be found in stereoisomers. Now, lipids themselves don't have a monomer like carbohydrates do and monosaccharides. Instead, they have many classes of lipids, each with their own components, such as triglycerides and phospholipids. They have fatty acid chains as their components. Depending on the presence of double bonds or not, that will affect their structure and function. Proteins are amino acids made up of a amine group, or carboxyl group, and then variable R groups attached to the central carbon and hydrogen. Nucleic acids are going to be your nucleotides consisting of pentose sugar, inorganic phosphate group, and a nitrogenous base. Now, the polymers of these different molecules are going to be your glucose, starch, glycogen for your carbohydrates, and lipids will have lipids. There are 3 classes. You have simple ones, which are the esters of fatty acids and alcohols. Most of these are going to be your sterols or your waxes. You have compound lipids, which are made up of fatty acids, alcohol, and y'all, whoever joining, like, be muted. So many people are joining unmuted. Stop doing that. But it's bothering me. An example of compound lipids are lipoproteins and glycoproteins. You have your derived lipids, which are basically derived from either simple or compound lipids. These are going to be your sex hormones, your bile acids, and cholesterol. And then amino acids form peptides. The whole thing with the folding creates complex structures and chemical properties. We'll get to that later on. And nucleic acids, they bind together to make complex polynucleotide polynucleotide chains for DNA and RNA. So a brief overview of metabolism is that metabolism is going to be the sum of all enzymatic reactions in a cell. This is made up of anabolism and catabolism. So anabolism is building more complex molecules from simpler molecules. Metabolism is building more simple molecules by breaking down more complex molecules. I associate the word catabolism with cannibalism. That's because my teacher told me to associate them together because cannibals break people and catabolism breaks molecules. So therefore, anabolism is going to be associated with condensation reactions, where you're going to be building these molecules. Hydrolysis is going to be associated with metabolism because you're using water to break molecules. Also friendly reminder, you should know the different types of bonds for these. Monosaccharides are going to be joining together with glycosidic linkages. Lipids are going to be joining together with ester linkages. Proteins are going to be joined together. Amino acids are going to be joined together with peptide bonds, and nucleic acids are going to be joined together with phosphodiester bonds. Now, there's also another component important to understanding the curriculum. They want you to know that urea is a compound that can be that is organic but can be artificially made. This is relevant because it falsifies vitalism. Because back in the 1800s, it was believed that only organic molecules can be made from living systems. Urea, which is waste from nitrogen metabolism, is made from nitrogen metabolism. Mammals excrete this via the kidney, but it is artificially made from heating inorganic ammonium cyanide to create urea, therefore falsifying vitalism. That's just a random part they just threw in, kind of, but that's also a standard you have to know. There are going to be a few drawings that you will have to remember. For the sugars, you are only expected to know the ring form, so you don't have to draw out the long chain. Don't do that. You're going to need to know your generalized amino acid for drawing. You don't have to know any of the different R groups. There are over 20. Please do not memorize them. And then you just need to know the general structure of a example lipid. You don't need to know the name. You just need to know this, draw this, you'll be fine. What else? On to the next one, 2.2. So water is an important molecule for biological functions because of its polarity. This polarity is happening because oxygen is more electronegative, so it's pulling on the electrons of hydrogen harder than hydrogen can pull them, so there's unequal sharing. That's why there's a net partial negative over here and two positive dipoles over here. So these hydrogen bonds are going to happen when hydrogen bonds to oxygen, nitrogen, or fluoride. And these are fairly stronger than other polar associations. And they increase the strength, the high specific heat, the boiling point, condensation, and adhesion. And it's why water is known as the universal solvent. So IB wants you to be able to compare water with methane. This is because water and methane have comparable size and electron geometry. Water itself has some lone pairs over here and methane doesn't, but they are similar electron geometry and comparable size because they're both tetrahedral and they have similar mass. However, the differences arise because of their polarity, because water has lone pairs, making it bent, and There's also oxygen's electronegativity. Methane is nonpolar, so it can only form some weak dispersion forces. Water, because it is polar, can form intermolecular hydrogen bonds, so different water molecules can bind with one another. Because of this, water has higher boiling points, melting points, higher specific heat, higher enthalpy of vaporization, and higher enthalpy of fusion. And these properties make it a great component of sweat, because sweat absorbs heat and prevents the body from overheating. This is good because the water in our sweat has a high specific heat, therefore it can absorb extensive amounts of heat before evaporating, making it an effective coolant. There's also going to be cohesion and adhesion forces. I don't think I actually made a slide for it, but you're going to have some surface tension with cohesion forces. Think of the water striders. And adhesion forces is with capillary action in plants. You're going to need to know transport in the blood and the differences between hydrophobic and hydrophilic molecules. Hydrophilic molecules means water-loving, so they freely dissolve and dissociate in water. These are going to be your polar molecules, your ions, glucose, amino acid, and oxygen. I know a lot of you might think, why oxygen isn't that carried in hemoglobin? Well, oxygen can be dissolved in the blood in low amounts, but most of it is attached to hemoglobin. And then there's also sodium chloride. Sodium chloride is an ionic compound, so it will actually disassociate completely into Na+ and Cl-. The negative parts of the water molecules will be facing the sodium, and vice versa for the chloride. So because they're polar, they can associate with the different charges. The hydrophobic ones don't dissolve well in water. These are going to be your cholesterols and fats that have to be transported with lipoproteins. When cholesterol and fats associate with proteins to make lipoproteins, they have hydrophilic sides that will face outward, and then the hydrophobic sides are facing inward, so they're shielded. That should be that. So next on to carbohydrates. So condensation reactions form glycosidic linkages. You're going to need to know sucrose, lactose, and maltose as important disaccharides. You should know their components too. Maltose is a glucose and a glucose, lactose is a galactose and a glucose, and sucrose is a fructose and a glucose. They're all glucose plus something else. Now on to the polysaccharides. So cellulose is a structural polypeptide, and starch is also a plant saccharide. Cellulose is a plant saccharide, and so is starch. So these are, where's my mouse going? So amylose and amylopectin are forms of starch. You'll need to know that. So as you can see here, cellulose is pretty linear because it has 1,4 glycosidic bonds, and the general shape and bond of cellulose makes it undigestible to most animals, including us. Cacotrophs and ruminants can digest them, but not humans. Starch, these two molecules are plant energy polysaccharides. Once again, they also have 1,4 glycosidic linkages, and they're also made up of alpha glucose subunits. Now, the difference between amylose and amylopectin obviously come from their shape. So the branching of these chains is because there is some 1,6 glycosidic linkages, Well, amylose only has 1,4 glycosidic linkages, so it's pretty linear in terms of its shape. Amylose is actually the preferred energy storage molecule of plants because, while it is hard to digest and is less soluble, the overall shape makes it easier to stack because it is just this linear straight shape instead of this branching shape. So it's more space efficient. And we have glycogen here. That's going to be our animal polysaccharide. This is going to be found in the liver, and it is made up of alpha glucose chains. And it has a mixture of 1,4 and 1,6 glycosidic linkages. So you can see that with the branching shape. With the type of bonds and linkages, it is similar to, it is analogous actually to plant amylopectin, just different frequency of, as far as I know, different frequency of 1,6 bonds. On to fatty acids. So fatty acids. Lipids are generally better at being long-term storage molecules just because of the way they stack and they're more stable. They're also far less easily digested than sugars, which are more short-term than anything. So fatty acids will have their large hydrocarbon chains found in phospholipid and triglycerides. You will have saturated fatty acids, which have no double bonds on either side of their molecules. You have monosaturated, which has at least you have monosaturated, which does have one double bond, and polysaturated, which has more than one. Furthermore, unsaturated fats can be cis or trans. This just depends on which side the double bond, the hydrogen of the double bond is. So if they're on the same side, it is going to be cis. If they're trans, it's going to be on the opposite side. Cis fats are the more natural one. Trans fats are usually synthetic. They're made through hydrogenation. They're very linear, even though they are unsaturated fats. And like saturated fatty acids, they're usually found solid at room temperature. So triglycerides are also another lipid family. They are the largest one. They're great for long-term storage. So they're created through the condensation reaction of three fatty acids and a glycerol molecule. These can be either unsaturated or saturated, but you won't have to know like how it is specifically unsaturated or saturated. These are found in animals as fats and oil in plants. So the IB does want you to know the health claims and risk associated with lipid consumption. Try to keep it simple because this is the one people usually haven't studied a lot on. So saturated and trans fats increase cholesterol levels, while cis and unsaturated fats decrease the cholesterol level. Fats and cholesterol will associate together in lipoproteins. Low-density lipoproteins, or LDLs, will carry cholesterol from the liver into circulation, so it raises the amount of cholesterol in the blood, while HDL finds the excess amount of cholesterol and brings it to the liver for disposal, so it lowers the cholesterol level in the blood. So saturated fats, they will increase the amount of LDLs and therefore increase cholesterol. Trans fats, they also increase LDLs, but they also decrease the amounts of HDLs. So it significantly increases cholesterol. So these are the ones that are the worst for you. Cis fats are generally considered good because they increase the amount of HDLs while lowering the amount of cholesterol in your blood. So high cholesterol can lead to atherosclerosis. You need to know that. That is the thinning and narrowing of bloodway passages. So it makes it easier for arterial blockages and plaques in your blood vessels to form. Eventually, this can lead to coronary heart disease, and then stroke and heart attacks. You'll have to know the health claims and evidence for lipid risk. So the claim is that food with high saturated and trans fats will increase Chd risk, and foods with high cis fats will decrease Chd risk. So the evidence for this is that there's a positive correlation between saturated fat intake and Chd. However, some populations don't fit this trend, such as the Maasai people. They have incredibly high saturated fat intake, but low amounts of Chd. A lower intake of saturated fats reduces the factors associated with Chd, such as obesity and high blood pressure. However, these studies can be flawed because size and cohort composition and how long the study goes on for are important, and their potential design flaws. Chd deaths are linked to high concentration of trans fats found in diseased arterial deposits. The other factors can play a role, such as genetics, and it has been found that blood cholesterol level has relatively weak association with your dietary standards. And lowering the amount of saturated and trans fats, so this is evidence against now. So lowering saturated and trans fats have been observed in Western diets. However, the rate of chronic heart disease has increased. However, this could be because Western diets are including carbohydrate intake far more. Odds are if you are eating anything, it might not even be sweet. It probably has high fructose corn syrup in it. We put high fructose corn syrup in literally everything. Like there's so much sugar in salads of all things and ketchup. So increased carbohydrate intake can lead to higher rates of diabetes and obesity, which are factors correlated and linked to Chd. So, and Chd is also something that is intersectional as an issue. It could be diet, it could be access to medicine, it could be genetics, it could be certain racial composition. Certain racial groups are more prone to Chd. This is particularly so with Native Americans that have high risk for diabetes. And the last part of this is BMI. So you just need to know how to calculate BMI. It's mass in kilograms divided by height in meters squared. Also know that BMI is not to be used in a vacuum. It should be used in combination with other metrics because there are going to be exceptions to BMI. This is true of like any statistic, you never use it by itself. BMI particularly doesn't work great with pregnant women or athletes. You also need to know how to use a nomogram, which is this over here, calculates BMI. The nomogram works, so say you choose a certain weight and then you choose the height. So then you kind of draw a line between the weight and the height, and then the value in between is going to be your BMI. And then you associate it with this chart over here. So 50 would be overweight if I'm doing this. That's just a quick thing. So we're going to be on to 2.4. So amino acids, they have 20 different R groups. You are going to need to know how a dipeptide forms. So you need to be able to draw a diagram of this. So these different side chains can give amino acids their properties, causing protein folding and function to vary based on the position in the polypeptide chain. Most polypeptides are around 50 to 2,000 monomers long. So organisms can create a large range of possible polypeptides. The possibilities are numerous. So let's talk about the most important part of polypeptides. So their 3D function is, the 3D conformation is going to be important. So the primary The primary structure is just going to be the regular order of amino acids. So this is going to be dependent on our group. And then you're going to have stable configurations of alpha helices over here or beta pleated sheets. These are going to be your coiled spiral shaped, and these are going to be more staggered. These are the results of hydrogen bonding between the amine group and the carboxyl groups. And then you have your tertiary structure, which is going to be the overall 3D shape determined by R group interactions, such as hydrogen bonding, disulfide bridging, polar and ionic bonding. And then some proteins will have a quaternary structure because some have more than one polypeptide chain. Not all of them do, but an example is going to be hemoglobin, which has many polypeptides to create the one functional protein. So I use the one from BioNinja just because like it says shits me, which is completely ridiculous. So you need to know the structure and function, not the structure, but the function for all of these. These are explicitly a standard, so you need to know these as examples and their function, except for these two. But these two, you will be expected to know them later on when it comes to movement, so it doesn't really matter, so you have to know all of these anyways. So you have your structural proteins like collagen and spider silk. Collagen makes up the bulk of the connective tissue. It is the most common protein in our human body. Spider silk, pretty self-explanatory, tougher than Kevlar. Insulin regulates and works with glucose levels in the blood. Different IGs, different forms of IGs have different effectivity against different pathogens, depending on the different proteins. Different IGs, there are 5 classes. I won't go more into that. Rhodopsin, which is going to be the pigment, which is going to be a pigment that is associated with perceiving light. Actin and myosin. Rubisco, which is going to be an important molecule for the light independent reaction. We will talk more about this for topic 8. Okay, let's see. Okay, you need to know also how denaturation works. This is going to be the unfolding and therefore function loss of a protein. It's usually permanent, and it's usually caused by pH and temperature changes. So temperature changes usually disrupt H bonding. And because human proteins are optimal at certain temperatures, it's going to be 37 degrees Celsius for most of them, almost all of them actually. Amino acids have different positive and negative regions, so pH alters their charge and therefore their solubility. So this is important when you're talking about enzymes for 2.5. So higher temperature means a faster reaction rate, and low temperature usually means an insufficient amount of energy to start reaction. If it's too high, it's going to cause instability, causing the hydrogen bonds to break. And when you have changes to the pH, the solubility and overall shape are going to be altered. And all enzymes have an optimal pH depending on their aqueous environments. The contents of your stomach, the proteins in there, like acidic environments, while the ones in your colon are going to be far less acidic. concentration is going to be the last factor that affects enzyme activity. So a higher concentration means higher activity. That means more chances to collide. But when the substrate concentration is highly saturated, the reaction rate tapers, and because all enzymes are occupied, you do need to know that this point where it tapers off is called Bmax. It's V subscript max. So this is the point where the enzymes are working at their highest capacity and they can no longer take more. You just have to wait for an enzyme to be finished, and then it will engage the next substrate molecule. So with enzymes, these are going to be globular catalysts. You need to know about the active site, which is where substrates bind to enzyme. And then we can also discuss collision theory. There are three conditions. They must meet to collide. They must do it with the correct orientation. So the active site has to collide with the substrate, and it has to have sufficient energy to cause the reaction to occur. The more frequent the collisions, that is when you have higher temperatures and concentration, that is going to cause more frequent collisions. With temperatures increasing, that's going to cause more kinetic energy. With concentration increase, that's going to be more chances to collide because things are so densely packed. The active site is also going to be largely determined by the tertiary site. So remember how we just discussed different changes to the pH and the temperature, how it can disrupt hydrogen bonding. And we talked about how hydrogen bonding is an important part of our group interactions from 2.4. So remember that, just kind of keep that in mind. And then we're gonna have industrial enzymes. So immobilized enzymes they're similar to membrane-bound enzymes in that they minimize random movement and they're more efficient. And also the separation of product is more easily achieved if the enzymes are static. Lactose products are going to be an important application of this. So treating milk with lactase. So lactase is extracted and purified from yeast and bacteria and bound to inert objects, inert substances like alginate beads. And the milk is run through over the enzyme repeatedly until it's completely purified. This is important because most people in the world are lactose intolerant. This is going to be the lowest in European and North American countries, but you produce a lot of lactase as you are born, and over time that concentration decreases into adulthood. So most people, by the time they're adults, they are some form of lactose intolerant. The rates can get pretty high, up to like 90% in East Asia and Southeast Asia. Also, it's relevant because lactose-free products are sweeter because monosaccharides tend to be sweeter. And this reduces crystallization too, because monosaccharides crystallize far less. And bacteria ferment monosaccharides better, so it's going to lead to a faster dairy product. I think that was actually relatively quick for everything. So yeah, that should be the end of this section.